all well. We're delighted to welcome Trefor Morris to today's webinar. What does whole genome sequencing reveal about anaerobic bacteria? Trefor is the Lead Biomedical Scientist for the UK Anaerobe Reference Unit at University Hospital Wales in Cardiff, where he plays a pivotal role in the investigation and characterisation of clinically important anaerobic bacteria. His work focuses on the laboratory analysis of anaerobic pathogens and the application of modern genomic tools to better understand these organisms. At the UK Anaerobe Reference Unit, whole genome sequencing is increasingly used to support the identification and surveillance of anaerobic bacteria, as well as the investigation of antimicrobial resistance and potential transmission events. Historically, techniques such as ribotyping were used to analyse bacterial genomes, but whole genome sequencing now provides a far deeper insight, enabling improved transmission tracking, uncovering hidden outbreaks, and enhanced monitoring of antimicrobial resistance. In today's session, Trefor will share insights from his work and explore what whole genome sequencing is revealing about anaerobic bacteria, and how these advances are helping to shape the modern analysis and understanding of these clinically important organisms. If you have any questions, please leave them in the chat box and we will answer them at the end. Thank you, and we hope you enjoy the webinar.
Good afternoon and welcome. My name is Trefor Morris. I'm the Lead Biomedical Scientist at the UK Anaerobe Reference Unit in Cardiff in Wales in the UK, and today I'm going to give you a talk on what whole genome sequencing teaches us about anaerobic bacteria. So what's coming up? I'm going to remind you about anaerobic bacteria and give you a brief summary of what we do at the UK Anaerobe Reference Unit. We'll go through the evolution of whole genome sequencing, where it started and where we are now. We'll talk about the impact of WGS on clinical diagnostics, and then I'll go through the impact that we're seeing within our own reference laboratory, which will be applicable to other reference laboratories as well, I'm sure. And then we'll go through some of the beneficial health impacts of anaerobic bacteria, where WGS has played a part in those sort of discoveries in terms of disease associations and a few other elements as well. The UK Anaerobe Reference Unit is primarily involved in the functions seen here. So we do identification, susceptibility testing and surveillance of anaerobic bacteria. We give clinical and technical advice. We do WGS surveillance for the whole of Wales on C. difficile. We also maintain a large collection of clinical isolates, which has grown to over 80,000 now. We're involved in research and development, and we also provide training and education via our 2-day practical course, Practical and Clinical Microbiology of Anaerobes, which I'll mention again a bit later. So if you are particularly interested in surveillance, we do have an open call at the moment for Bacteroides species. We last did a full surveillance on Bacteroides way back in 2016, and we're aware of an increase in resistance being reported in other countries. So please do scan the QR code if you're based in the UK lab and get involved. Just a quick reminder then about anaerobic bacteria. They were first discovered by Louis Pasteur in 1861 to 1863 and described as life without oxygen. And then in 1877, Pasteur and Joubert isolated the first pathogenic anaerobe and named it Clostridium septicum, and that's Clostridium septicum on an FAA plate that you can see on the slide there, which swarms all over the plate and hemolyzes it, which is an indication of how pathogenic that organism is. We mustn't forget that anaerobic bacteria include a diverse variety of organisms, which includes spirochetes and methanogens, and they cause disease in both humans and animals, and very similar diseases in some instances. For example, tetanus, which is caused by Clostridium tetani, and these infections are often polymicrobial, and that makes them quite complex, and they also involve interactions between anaerobes and aerobes. For example, in chronic sinusitis, where Staphylococcus aureus is also involved. Anaerobic infections are caused by endogenous microbiota, and they cause infections when there are predisposing factors in play, such as compromised blood supply, trauma, any foreign bodies or blood clots. And these are the organisms or the genus that are involved at various different bodily sites. It's not a comprehensive list. There are many more involved, but what I'm going to do is take you through the main pathogens. The main anaerobes that cause human disease are grouped into these three genus, so Clostridium, Bacteroides and Fusobacterium. From the Clostridium, you can further split them into three different groups. So there's a neurotoxic group, Clostridium tetani and Clostridium botulinum, the cause of tetanus and botulism. And then we have the histotoxic group, which cause gas gangrene, so perfringens is the most common, but also C. septicum, Sodeli and Novii, which are rarer causes of gas gangrene. And then we have the enterotoxic group, Clostridioides difficile and Clostridium perfringens. And within the Bacteroides, I would say that the Bacteroides fragilis is our primary pathogen, and that causes intra-abdominal infections and bacteremia, as do other Bacteroides species. And then Fusobacterium, we have two main pathogens, Fusobacterium necrophorum, which is the primary cause of Lemierre's syndrome, and Fusobacterium nucleatum, which is often seen in dental infections. Sequencing has evolved hugely over the last 50-60 years, starting way back in the 1950s when DNA structure was first understood following the work of Rosalind Franklin and then the publication of the DNA double helix by Watson and Crick. And then a decade later, DNA sequencing, the basis of sequencing DNA was published by Frederick Sanger. During the 70s and 80s, we had the first Sanger sequencers being commercialized, and then we've moved on in the 90s to 2000s with the second generation or short read sequencers that are commonplace now, and then further into the 2010s and the 2020s. We see the long read sequencers and true WGS coming into play. In terms of the impact that sequencing is having within clinical laboratories, this publication by Hasmon and colleagues from 2014 summarises it very well. So in the first diagram that they have within the publication, it shows a very complex workflow culminating in typing, which could take a number of weeks, whereas in a genomics diagnostic pathway, which is still dependent on culture, the turnaround time is significantly reduced, and the information that you're able to glean within that turnaround time of a number of days is superior to the traditional methods. You can have comparison with global databases and typing information available in a very short period of time. And we are rapidly moving towards use of WGS directly from samples, and the impact of that is huge in terms of turnaround time. So we are talking less than a couple of days to all the way from the sample arriving in the laboratory and WGS being performed and all of the analysis being available, so a huge potential impact. Moving on to the impact on us as a reference unit. I'm summarising these in 5 different areas. So the first area I'll cover is new species identification and reclassification of current species as well. And then there's bacterial typing improvements, some of which have impacted us hugely in the Anaerobe Reference Unit. There's the impact on detection of antimicrobial resistance and the surveillance that we do as well. And then there's the question of phenotypic versus genotypic resistance detection, and I'll mention a little bit about horizontal gene transfer as well. In terms of impact in terms of new species, you can see here one example of the discovery of Olsonella lacoscia from feral chickens. For those of you who don't know what a feral chicken looks like, and there's a picture there. And then there's new species, and again this is just one example of many that are available in the literature. This is Anaerococcus campinii from the Dutch group that Linda Blue leads, and here they've discovered a new Anaerococcus from their processing, which is similar to ours, of using the MALDI-TOF initially, and then 16S sequencing, and then for those that they see consistently from certain infections, they've managed to associate together and discover the new and name the new species. So in terms of reclassification of bacterial species, probably the most famous anaerobic bacteria has been reclassified recently. It's Clostridium difficile, as it was then known. And Lawson and colleagues put this publication together a number of years ago that clearly demonstrated that Clostridium difficile didn't belong within the Clostridium group, and thankfully they proposed the name Clostridioides difficile, which was very different to what had been proposed from a number of other sources over the years up to that point. So thankfully you can still call me C. diff. In terms of more recent reclassification, it's been recognised for a number of years that Bacteroides fragilis can be divided into 2 very different bacterial species. So there's a division 1 and division 2 within Bacteroides fragilis, and they have very different genomes, and we are aware that they are distinct species. One has a cephalosporinase, division 1 called CEP-A, and division 2 has a carbapenemase, CFI-A, and you can clearly see differences in their carbohydrate metabolism from the gene composition, which is highlighted in that publication. In terms of the second major impact on reference laboratories typing, we can see very well documented and published schemes for typing of Bacteroides fragilis and Clostridium perfringens that have been designed via whole genome sequencing, and these are available now to be utilised. In terms of the impact of typing within the Anaerobe Reference Laboratory, we have moved on from using PCR ripe typing to using whole genome sequencing for our Clostridioides difficile strains that we culture from across Wales, and this process is highlighted in this diagram here. It's the first part of it is done obviously in the clinical laboratories. That takes 4 to 24 hours, and then another up to a week for the process to be cultured in our lab, and then for the DNA sequencing, the whole genome sequencing to happen within our pathogen genomics laboratory, and we end up with a cluster code assigned to strains that are related, and the whole process now takes 7 to 10 days. The majority of them are actually sequenced within and to result within a week. So that's a huge impact within our reference laboratory. For those who are particularly interested in the details of that process, it's highlighted in this second slide here. So this is our WGS SNP typing pipeline. All the details are there. We use multiple reference strains, which is unique in terms of SNP typing pipelines. The software is open source, and we assign clusters at the 5 SNP level, and the process is being validated and is also UKAS accredited. The third major impact on us as a reference laboratory and other laboratories is the detection of antimicrobial resistance and the evolution of that resistance. So I've just highlighted a few publications here. The first is discovery of a novel antimicrobial resistance gene, metallobetalactamase, in Bacteroides xylanisolvans, which has a lot of similarities to the CFA of Bacteroides fragilis, and also needs insertion elements for its activation. And the second I'm highlighting is one describing the evolution of resistance in Bacteroides nordiae. This was a few years ago now, when there was a paucity of data in terms of Bacteroides nordiae, and they used whole genome sequencing to look at this particular isolate, and discovered that it was harbouring the NIM-E gene, which is thought to be involved in metronidazole resistance. And then the third publication here, as an example, is one from Dr. Michael Perry from the Anaerobe Reference Unit, and he has performed the first large-scale study of almost 400 strains of Fusobacterium necrophorum, and concluded that continued surveillance is necessary, as we discovered a transposon-mediated beta-lactamase resistance determinant in those strains. And whole genome sequencing has revealed a huge amount about Bacteroides fragilis. It is probably our major anaerobic pathogen, including the detection of an international multi-drug resistance Bacteroides fragilis cluster, and the unveiling of some of the insertion elements that are required to turn on the resistance for meropenem and metronidazole, and there's been numerous plasmids that have also been identified as well through the use of whole genome sequencing. The fourth major impact on reference laboratory microbiology for anaerobes is the comparison of phenotypic and genotypic antimicrobial resistance detection and we're not quite there yet in terms of really saying we can replace phenotypic with genotypic methods, but it's something that we're continually looking at. And UKAS published a number of years ago along the lines of saying that we're not there yet, and we're continually working with UKAS using our data from our agar dilution AST methodology to work through new breakpoints and look at whether within the wild type distribution any resistance determinants are present. So I would say that's a work in progress. The fifth major impact on reference laboratories is the determination of horizontal gene transfer. And I've just highlighted a couple of publications here, the first of which indicates distinct dichotomy between anaerobes and aerobes in terms of their beta-lactamase genes, and then the second here indicates the transfer of a very similar CFI element between two Bacteroides species within a sample, and the key significance there is that obviously if that's happening within a sample, then that is very important and worrying. I know there's further work ongoing looking at more samples as part of the European study group that I'll mention in a short while. So moving away from the impact on the laboratory, just to go through a few things that whole genome sequencing has shown us in terms of beneficial health impact. So colonization resistance is a term that we see more and more frequently in the literature, and in terms of anaerobic bacteria, the short-chain fatty acids that they produce are critical components in conferring this colonization resistance, and that allows them to confer resistance against pathogenic bacterial colonization. And whole genome sequencing reveals key differences between different species involved or supposedly involved here, and supports the selection of certain strains versus others for use as probiotics. So this is also well studied in animals. I'll just give you a couple of interesting examples. The first is Cetobacterium, probiotic potential in Nile fish, and the second is in the marmoset monkey and its Bifidobacterium, which plays a critical role in carbohydrate metabolism, and these things again are being understood better because of whole genome sequencing of strains. Moving away from the benefits that we've found from whole genome sequencing strains, anaerobes are more and more highlighted as being involved in various different diseases or associated with different diseases, and I'd recommend you have a look at this really nice summary paper from Bionova and colleagues, and they've detailed here some associations that have been put together more recently, and I'll highlight a few in the following slides, but all of it is supported by being able to perform whole genome sequencing. To highlight a few of those disease associations from the previous publication, one example is Akkermansia, bifidobacterium and association with obesity, where a reduction is seen in Akkermansia and bifidobacterium, which is linked to weight gain, and Akkermansia itself is seen to impact on epithelial barrier function, but this is not 100% beneficial in terms of Akkermansia, as there are also publications demonstrating an increase associated with children with eczema. The second association I'd like to highlight is between Faecalibacterium prausnitzii and inflammatory bowel disease, and this association is quite strong, and publications suggesting that it might actually serve as a biomarker of the disease. And Faecalibacterium prausnitzii is one of the main butyrate producers in the intestine and makes up about 5% of the total fecal microbiota. It's a very, very difficult anaerobe to cultivate, as it's extremely oxygen sensitive, and then WGS is being used to determine the characteristics of newer species, and there's a really nice mini review that I've highlighted there if you would like to know more about that particular association. The association I'd like to highlight is that between Fusobacterium nucleatum and oral and colorectal cancer, and there's more and more information coming out year on year about this. So Fusobacterium nucleatum is a key pro-carcinogenic bacterium, and we see significant overabundance of this particular anaerobe in tumour tissues, with high levels associated with very poor prognosis. There's a really nice publication there detailing Fusobacterium's involvement in periodontitis and subsequently its association with colorectal cancer. The fourth disease association I'd like to highlight is the association of anaerobes with bacterial vaginosis, which we've known about for many years. However, whole genome sequencing is doing a number of things, revealing newer species which are better characterized by whole genome sequencing, and also revealing that metronidazole treatment might not be the most appropriate in all cases, and that we should be informed more by the WGS data in terms of managing patients with bacterial vaginosis. The final disease association I'd like to highlight, which again we've known about for many years, is the role of Porphyromonas gingivalis in dental disease, which WGS is showing us more and more the importance of a shift in the diversity of microbial communities, and also that this may impact on other inflammatory disorders, and it's demonstrated to us that there are extensive gene rearrangements in different strains of Porphyromonas gingivalis, which may be significant in terms of their potential to cause disease. Whole genome sequencing is also teaching us more and more about the industrial applications of anaerobic bacteria, and there's a wide range of these. I've just selected a few here. There's producing electricity, and there's the breakdown of pollutants, and there's the conversion of starch to bioethanol, and the photosynthesis or photosynthetic properties of certain anaerobic bacteria as well, so to name but a few. Summary then, in terms of anaerobes and whole genome sequencing, we are very much still learning. Whole genome sequencing is driving forwards our understanding of the role of anaerobes in health and disease, and in industrial applications. In the laboratory, the key thing to highlight is don't assume that anaerobes are susceptible, and please perform the UKAS disc susceptibility testing, which is now available, and whole genome sequencing is telling us that multi-drug resistance exists, particularly in bacteroides species, so it's something to look out for. And that brings me to the end of my talk. Thank you very much for listening, and please do get involved. I'd particularly like to highlight the work that we do as part of the European Study Group for Anaerobic Infections, SCI, and the upcoming webinar on metronidazole resistance that you can link to via the QR code displayed there. And the second shameless plug here for our practical and clinical microbiology of anaerobes course, which is a special one this year for two reasons, it being the 50th anniversary of Don Whitley Scientific, as well as the 10th anniversary year for us running the course together. So please scan the QR code if you are interested. I'd like to finish by thanking my colleagues from the Anaerobe Reference Unit, so in that picture there, as well as other colleagues from across Wales, from Public Health Wales, Cardiff University, and colleagues from Pathogen Genomics Unit as well. And of course to Don Whitley Scientific for the invitation to speak on this webinar series. So I'll finish by just leaving you with a quote that a colleague gave us a number of years ago.
Well, hello everyone, and thank you very much for joining our webinar today. I hope you enjoyed that presentation from Trevor. Trevor is live with us now, and he'll be able to take any questions that the audience might ask. Now, we usually find that we get a rush of questions after the talk is finished, but we do have a few up already, so if you're ready there, Trevor. First, we have, when using whole genome sequencing for speciation, are there distinct species or are there intermediate genotypes which sit in between?
Yeah, so we're learning an awful lot through using whole genome sequencing about different species that sit within. I gave a couple of examples there, and certainly the knowledge is increasing, month by month, year on year. So we've obviously done a lot of work with Bacteroides fragilis, and we can see that that's definitely two different species. We haven't successfully, I think, renamed that yet, but I'm sure that'll work its way through. And I know there's lots going on in the Clostridium genus as well. Lots of what we know as Clostridium doesn't fit, and lots going on with Fusobacterium nucleatum as well. So yeah, we're learning more and more every month.
Yeah, sure. And of course, Clostridium is going to be quite a diverse genus, I think, because of the way they were classically categorized. They were anaerobic spore formers, and we find Definitely. That Clostridium and with Bacillus too, of course. So, yeah, from my own point of view, we do a lot of susceptibility testing here, and you mentioned the UCAST AST work. Are we yet able to perform any susceptibility testing against anaerobes using discs?
Yeah, absolutely. So we've already got a joint publication with UCAST highlighting some key species that you can test. We can, and that's been there for a couple of years now. So we focused on ones that were relatively straightforward to grow and would grow in pretty well in 24 hours. So we've got Bacteroides species, Prevotella species, we've got Fusobacterium macrophorum, Cutibacterium acanes, and Clostridium perfringens already part of that piece of work. So all the details for those are available on the UCAST website, and we're working with UCAST as we speak. Actually, my colleagues are working on this. Dr. Sarah Kopsi-Mauer, who gave a talk earlier in this webinar series, is working hard behind the scenes to provide some further details and data to allow us to expand the number of anaerobes that's covered by that disc testing criteria. Please do go and have a look on the UCAS website. It's a fantastic piece of work, something that hasn't ever been formally acknowledged and developed in this way. So very much something we're very, very pleased about.
Yeah, thank you. Excellent news, which will have a real impact on the practicality of susceptibility testing for sure. Moving on then, how has whole genome sequencing impacted infection control practices?
The easiest one to start with there, I guess, is with Clostridioides difficile. So this has had a huge impact in terms of IP&C practices in terms of managing C. diff outbreaks. We've had whole genome sequencing SNP analysis available to practitioners within IP&C in Wales for 3 or 4 years now. And the way that we run that service means that they can impact on outbreaks and manage them much more effectively in much closer to real time. So getting results within sort of an average of 7 days is fantastic for them in a positive and negative way, if you understand what I mean. So it's equally as valuable to be able to rule things out of an outbreak as it is to be able to rule them in and focus your investigation in the right way, because we're very conscious that within IP&C the pressures on time are huge. So if you can make sure that any targeted learning as well is focused on where an outbreak started and not where you've managed to pick it up, it's also hugely valuable, but extremely, much more complicated in terms of C. diff as a spore bearer. We know that is challenging us to think very differently in terms of where an outbreak starts and how long a duration there is between somebody maybe picking up a strain and then actually becoming symptomatic and being part of an outbreak. It's having huge impact, I feel, in IP&C.
Sure, thanks very much. So moving on, how much of an impact do you think sequencing bias has on identifying and understanding resistance genes in anaerobes?
Oh, that's a tricky one, actually. We only use one sequencing platform, and we're not we're not able to really drill down into that at the moment. I don't think it's in enough kind of common use and to be able to really answer that question fully yet. But again, it's it's happening more and more, and it's something that we're learning more and more about.
Okay, thank you. Moving back to AST, what steps are you doing to elevate genotypic AST?
So yeah, again, we working with UCAST, improving the methodology for the disc testing. We're aligning what we're doing. So what we do in the background is perform agar dilution, which is the gold standard method for AST, and then we're marrying that up with the genotypic work that we're doing as part of our service development. We're not performing whole genome sequencing on everything, but anything of interest, we are phenotypically resistant or unusual. We're performing whole genome sequencing on, and then we're feeding that information back to inform the UCAST team, and then as we gather that data, we'll be able to make better decisions in terms of whether there is an opportunity to use WGS as a substitute for phenotypic AST in the future. But we're, as I said in the talk, we're not there yet, but we are working on it in the background.
Okay, thank you. Yeah. Next, we have somebody is wondering if there are any cases of anaerobic infections where the anaerobes are not in high abundance.
Yeah, I'm sure there are. We obviously run a reference laboratory, so it's not a clinically. We don't deal with clinical cases, but when we have cases, when we have orgs referred to us, we do get limited clinical information about the infections themselves, and so yeah, I mean they're almost always polymicrobial in nature. So you can imagine that they will be there in various different amounts. But obviously, that you do have the monomicrobial infections as well. So it's difficult for us to really make a defined determination of the amount of the
Sure.
the anaerobes in a particular infection.
Yeah, of course. Can you give a general guideline when to perform whole genome sequencing on a clinical anaerobe?
That's something we're working our way through at the moment. So I think we focus a lot on Bacteroides. Bacteroides species, Bacteroides fragilis, particularly. Bacteroides fragilis is, it's our most common pathogenic anaerobe in monomicrobial and in bacteremia. So, I think we would definitely focus our efforts on Bacteroides, and then I would say anything, if you're doing phenotypic testing and you have something unusual, something that maybe it doesn't make sense, if you like, so something that may have an odd resistance pattern, then you'd want to try and understand what's behind that a little bit more with whole genome sequencing.
Yeah, of course. Thank you. Which clinically important anaerobe is studied the most is a question we have here.
Bacteroides fragilis is one of them, I'm sure. I would say that's the most published about. There's a lot of interest in in those ones that make up the gut microbiota at the moment, and because of the links to various diseases that I've already mentioned, and a lot obviously of interest in C. diff still. C. diff still causes a huge amount of morbidity across the world, and a significant mortality. So yeah, C. diff is definitely up there too.
Okay, and following on from that, any upcoming anaerobes that you're seeing in high volumes that you haven't perhaps seen before?
I don't think we've seen a huge sort of. There's nothing springs to mind in terms of things that we've not seen before. So no, I can't think of anything. I mean, we're certainly understanding more because of the mouldy, and we're identifying more anaerobes more regularly. So I would say, previously you may have sent, a gram-positive rod out as a anaerobic coriniform. We're actually able to, to speciate a lot of those. Lots have gone into the Actinomyces genus. So we're learning a lot more that way. But I don't think there's anything that I could put my finger on and go, well, actually, we've seen an increase in people referring this.
Yeah, of course. And a simple but quite topical question: do you use any AI applications in your work?
Not at the moment, no. But I'm reading more and more about the use of AI and machine learning, predicting AST, etc. So it's really an exciting field to be keeping an eye on.
Yeah, of course. I can see the relevance and how it might work. Yeah. What is known at the moment on antimicrobial resistance in anaerobes relevant in dental infections?
Yeah, we're quite looking kind of to be linked in with a small microbiology lab that deals with dental infections across the road from us. And I think Dav, who works across the road, is very keen on AMR in dental infections, and particularly the increase in penicillin resistance within that group. So I think that's probably the major concern. And of course, in terms of dentistry, there's another concern as well in terms of the antimicrobial prescribing. In terms of potentially being an issue or a trigger for C. diff infections. I certainly feel there's not quite the same scrutiny over prescribing in the dental field. So you could see how and why potentially AMR would go, not as well monitored. But it's something that we're working on, and we're very keen to support.
Yeah, of course. There's a longer worded question here. I'll paraphrase it. Given the issues with culturing anaerobes, have you assessed the possibility of determining antimicrobial resistance directly in clinical samples such as blood from patients with anaerobic infections?
We haven't yet, but it is on our list to look at. Something that I know Michael Perry, who's also given a talk in his webinar series, is very keen on. He has a very strong molecular background, and I know direct PCR was was available in different labs, or has been for many years, and it's something that we're working on with our Pengu colleagues as a future project, whether we can look at direct whole genome sequencing from samples. I think it would be amazing, an amazing step forward again. Yeah,
for sure. Well, no further questions at the moment, but in case any late ones popped up, I'll take this opportunity to do my own shameless plug, to borrow your phrase, which is I think will be of interest to the audience, which is be about our next webinar. we're increasing the frequency of these anaerobic webinars, and our next one will be on antibiotic resistance mechanisms in anaerobic bacteria, their origin and implications, and that will be from Linda Bailu, who you mentioned earlier, University Medical Centre Groningen, and that will be on the 4th of June this year at the same time. So I'd encourage the audience to sign up for that. And I think while I've been speaking, it's been one more question for you, which is, can we use WGS to elucidate resistance mechanisms in bacterial vaginosis-associated bacteria such as Gardnerella vaginalis? Can we do that directly from the sample?
I don't think we can at the moment.
Yeah.
I think we still have to culture, which is one of the major challenges.
Yeah,
but it's very much, coming soon in the next few years, I would imagine.
Yeah, brilliant. I think that's it. My colleagues have posted the link for our next webinar up there in the questions. So thank you again to you, Trevor, and thank you to everyone else who signed up to participate. We will, of course, have recorded this and it'll be available. We've had a good sign up today, and I hope that everyone has enjoyed it. Thanks again, Trevor.
No problem.